Good day, my dear students. Once again, I want to welcome you to class of BCH 408. Our lecture series is number 3, and the topic is chemistry of oxygen and carbon dioxide transport. As usual, let's run down the objectives of this class. At the end of this period, you should be able to describe how oxygen binds to hemoglobin and the factors influencing the process. You should be in a position to explain the 3 primary methods for carbon dioxide transport in blood. You should be able to analyze the relationship between oxygen dissociation curves and physiological conditions. 4. Connect gas transport to acid-base balance through the bicarbonate buffer system. Now, before we go into details proper, let's look at definitions of certain terms. We have 3 we'll consider, that is oxygen, hemoglobin, and hemoglobin binding. Oxygen is a colorless, odorless gas that is essential for life. Hemoglobin, on the other hand, is an iron-containing protein found in red blood cells that plays an important role in the transport of oxygen from the lungs to tissues of the body. Now, what is hemoglobin binding? It simply refers to the process by which hemoglobin reversibly binds oxygen molecule to transport them from the lungs to tissues in the body. Now, let's look at oxygen transport. We just mentioned hemoglobin binding. Now, heme binds to oxygen, to a molecule of oxygen. This is the structure of heme as shown here before you in structure figure A. Figure B is the hemoglobin structure. Now, I'll come to that shortly. Now, heme as a molecule binds a molecule of oxygen, that is O2. Now, look at the structure B before you. You can see HB representing hemoglobin and 4 molecules of oxygen. Because a hemoglobin is a tetrapeptide molecule, it requires 4 molecules of oxygen. Remember that hemoglobin is made up of 4 sub-chains or sub-units as alpha 1, alpha 2, beta 1, and beta 2, as you can see. Now, each subunit will bind 1 molecule of oxygen. So, we are talking of 8 atoms of oxygen. That is 1 subunit binding 1 molecule. Remember that 1 molecule of oxygen is O2. So if you multiply the 4 by 2, you have 8. That explains the equation we have above each of the structures. Now, having understand the logic behind the equation, let's look at the binding proper. The reaction between oxygen and hemoglobin is reversible, which means that hemoglobin can bind to and release oxygen depending on factors like oxygen partial pressure, pH, and temperature. In tissues with low oxygen, the equation shifts left, and in lungs with high oxygen, it shifts right, binding oxygen, while in the former, it is releasing oxygen. Now, let's look at the nitty-gritty of hemoglobin binding. First of all, we look at the cooperative binding of hemoglobin. What does it mean? When one molecule of oxygen binds to hemoglobin, it changes the shape of the structure slightly, increasing the affinity of the remaining sites for oxygen. Now, hemoglobin this time, not hemoglobin. Remember, there's a difference between heme and hemoglobin. To have a full molecule of hemoglobin, you need 4 molecules of heme to form the subunits where proteins are attached later. This is crucial for efficient oxygen loading. When we talk of oxygen loading, we are referring to the binding of oxygen to hemoglobin, while unloading refers to the release of oxygen from the same hemoglobin. Oxygen dissociation curve. This is defined as a graph which shows the relationship between the partial pressure of oxygen and the percentage saturation of hemoglobin with oxygen. Now, if you look at the graph before you, the sides are named oxygen tension, the axis oxygen tension. You have percentage saturation of blood with oxygen to 100%. This graph is sigmoidal, meaning S-shaped, with each regions having a specific meaning. Now, let's consider the meaning. If you look at the low partial pressure, the middle or the median, and the high partial pressure, let's get to the definitions. Let's do a little bit of explanation on the phases of the curve. At low partial pressure, such as in resting tissues, hemoglobin affinity for oxygen is low. Oxygen binds slowly because the first molecule is hard to attach due to hemoglobin's initial structure. This ensures that hemoglobin can release oxygen where it is most needed. In the median partial pressure or the median section, small decrease in partial pressure of oxygen cause large oxygen release, ensuring adequate oxygen delivery to metabolically active tissues. That is, when the first molecule or when the first oxygen molecule binds, hemoglobin undergoes a conformational change, increasing its affinity for additional oxygen. This results in a steep increase in saturation with small increases in partial pressure of oxygen. In tissues with moderate oxygen levels, a small drop in oxygen partial pressure leads to a large release of oxygen, supporting metabolic activity. At high partial pressure of oxygen, hemoglobin becomes nearly saturated due to high oxygen affinity, enabling efficient oxygen loading. That is, at high oxygen pressures, hemoglobin is almost fully saturated. Further increases in partial pressure of oxygen can cause only slight increases in oxygen binding. This plateau ensures that hemoglobin efficiency picks up oxygen in the lungs. Now, carbon dioxide transport in blood. Three mechanisms are involved that are used or employed to transport carbon dioxide or carbon dioxide in the blood. And these mechanisms include the bicarbonate buffer system, the carbaminohemoglobin system, and the dissolved carbon dioxide mechanism of transport. We shall explain or take this in turns. Starting with bicarbonate buffer system. First of all, let's look at the carbon dioxide production in cells. Cells produce carbon dioxide as waste product during metabolic from 1. Electron transport chain and tricarboxylic acid cycle. Of course, you are familiar with this. Now, the carbon dioxide diffuses from tissues into blood. Inside the red blood cells, it reacts with water forming carbonic acid, as shown in the equation below. You can see it. And that reaction is catalyzed by carbonic anhydrase inside the red blood cell. Now, this figure or diagram is a representation to simplify the happenings in the red blood cells. Now, this red portion is the red blood cells. Now, inside the red blood cells, you have the bicarbonate ion as well as the hydrogen ion. Now, when carbonic anhydrase condenses carbon dioxide and water within the red blood cell to form carbonic acid, that compound immediately dissolves or decomposes to give bicarbonate ion and hydrogen ion. Now, the bicarbonate ion found its way out of the cell into the plasma, leaving the hydrogen ion. The hydrogen ion in turn combines with hemoglobin to form the reduced hemoglobin, which is taken into the plasma. Now, the reduced hemoglobin prevent acidification of the blood. Meanwhile, as bicarbonate is leaving the cell, it is replaced with chloride ion. Now, it is the movement of the chloride ion into the red blood cell that is referred to as the chloride shift. The chloride shift occurs to maintain electrical neutrality of the red blood cell. Still on the bicarbonate buffer system, what happens in the lungs? Because recall that the bicarbonate ion goes out of the blood cells into the plasma. Now they are carried to the lungs. What now happens? What happens is that the entire process is reversed, such that oxygen binds to hemoglobin, releasing hydrogen ions. Of course, you know that in the lungs, oxygen tension is high, or oxygen pressure is high. So it will bind with hemoglobin and release hydrogen ion. Hydrogen ion will in turn combine with bicarbonate ion to form carbonic acid. Then carbonic anhydrase then converts the carbonic acid into carbon dioxide and water. That is the carbon dioxide that is expelled through the lungs. The second method of carbon dioxide transport is Carbaminohemoglobin. Carbaminohemoglobin is a product formed by the reaction of carbon dioxide with hemoglobin, as indicated in the diagram before you. It's a reversible reaction. Carbaminohemoglobin is formed when carbon dioxide binds directly to hemoglobin. This binding occurs at the N-terminal end and amino acid side chains of amino acid such as arginine and glycine on the globin chains of hemoglobin molecule. Again, let's consider the transport mechanism. Carbon dioxide binds hemoglobin, forms a product called carbaminohemoglobin, which reduces hemoglobin affinity for oxygen. That phenomenon is referred to as the Bohr effect. Now, the Bohr effect facilitates oxygen release in tissue. When oxygen is released, it becomes deoxygenated. So, deoxygenated hemoglobin, which has a higher affinity for carbon dioxide, binds the gas in tissues and release them in the lungs. That is the Haldane effect. So let's look at the Bohr effect and the Haldane effect. What do they stand for and what is their similarity? The Bohr effect in tissues enhances oxygen loading. The Bohr effect in tissues enhances oxygen unloading. What I mean by oxygen unloading is the release of oxygen, while the Haldane effect ensures efficient carbon dioxide uptake. As oxygen is unloading, carbon dioxide is replacing it. Secondly, in the lungs, oxygenation improves oxygen loading because the partial pressure of oxygen is higher at that region, while Haldane effect promotes carbon dioxide expulsion. Thirdly, we must recognize that both effects are critical for maintaining acid-base balance and optimizing gas exchange under varying metabolic conditions. This is a summary table of the Bohr's effects as well as the Haldane effects. Now, we see here the primary focus, the trigger, the site of action, and the impact on hemoglobin. That is, Bohr's effect versus the Haldane effect. In both cases, the primary focus for Bohr's effect is oxygen release in tissue, while Haldane effect is carbon dioxide or hydrogen ion transport and release. What triggers Bohr's effect is high carbon dioxide concentration and low pH, while that of Haldane effect is low oxygen saturation. The site for action for the Bohr's effect is peripheral tissues, while the lungs and tissues is the site of action for the Haldane effect. Now, impact on hemoglobin reduces affinity, and for the Haldane effect increases carbon dioxide and hydrogen ion bonding capacity. Again, finally, let's look at dissolved carbon 4-oxide transport or dissolved carbon dioxide transport. Carbon dioxide is more soluble in blood than oxygen, and so it diffuses from tissues directly into capillaries and dissolves in the plasma due to its concentration gradient. Importance of dissolved carbon 4-oxide or carbon dioxide. 1, it contributes to blood pH regulation. 2, it is directly involved in stimulating the respiratory centers in the brain through pH changes. And 3, it serves as a reservoir for conversion to other forms like bicarbonates. Now, in conclusion of this lecture, we can see that gaseous exchange is essential for cellular metabolism and acid balance hemostasis. 2, that hemoglobin properties, that is, the bicarbonate buffer system and the interplay of the Bohr and Haldane effect ensures that oxygen is delivered to tissues and carbon dioxide removed. Finally, that these phenomena work together in maintaining stable blood pH. Having said that, I'll consider this point as a convenient place for me to stop this lecture today. For your study questions, take a look at these 5 questions and practice them very well. On a final note, I want to thank you for watching and listening. I wish you a happy day ahead.